Our last serological assays to talk about are the immunoassays. So again, these are immunologic methods, serological methods, and these are the most sensitive of all the methods. So there's two main immunoassays. We could either use a radio immunoassay or an ELISA. ELISA stands for enzyme-linked immunosorbent assay. So radio immunoassay is basically the same thing as ELISA, only ELISA, we're looking for a color change. That's how we can measure the results. Whereas for radio immunoassay, we're using radioactive isotopes, so we're looking for radioactivity. We would use a radio immunoassay for extraordinarily sensitive testing, like if there's very minute amounts of antibody or antigen, maybe for certain viruses. Whereas ELISA is way more commonly used. ELISA is probably one of the most common diagnostic tests other than maybe PCR. This would be the go-to test for the majority of more well-known and well-characterized diseases, such as, for example, HIV. ELISA is the first test used to screen for HIV, whereas I told you Western blot is the second test used to confirm a positive ELISA. What makes ELISA so great is that it's very quick, it's relatively simple, and you could do many samples at the same time. So if you had multiple patient samples to test, you could test multiple samples. Although it is possible to get false positives or negatives with ELISA, it's not as common as with some of the other methods we've talked about. And also ELISA can measure very minute quantities of antibodies, so it's extremely sensitive. So there's two different types of ELISA. We'll talk about both of them. There's indirect ELISA or capture, also known as sandwich ELISA. So ELISA utilizes these plates. These are called microtiter plates. And you can see these plates have 96 little holes in them. These are called wells, and that's where you insert the sample. So as you'll see in a minute, when you do see a color change, for example, in this test, it's yellow, but it could also be purple, blue. The color varies. So when you do see a color change, that indicates a positive result. And when you do not see a color change, it indicates a negative result. So let's talk first about the indirect ELISA. Indirect ELISA, like I mentioned, is the one we use for screening for HIV. It's the first test used anytime someone gets an HIV test. This is the one they go to. So here are the steps for indirect ELISA. In the first step, we're going to add the known antigen. So these are commercially available antigens that we have extracted from the pathogen. So for example, in this case, we'll say we are adding the HIV antigen. What will happen is the HIV antigen will stick to the walls of the plate. So in these little holes, these antigens will stick to the wall. In the next step, we're now going to add the patient's antibody. If the patient has antibodies that recognize HIV, that will bind to HIV, they'll bind. So for example, in sample A, this patient did have HIV antibodies, so they will stick to the HIV antigen. Whereas sample B, there was no HIV antibodies, so nothing would stick to the HIV antigen. The next thing we have to do is we have to wash or rinse out the plate to get rid of any leftover residual waste. Okay, so after you wash the plate, again, sample A, you still have the antibody bound to the antigen. But sample B, now you have washed away all of these other antibodies. They weren't HIV antibodies, they were other ones. You're going to rinse those all out. So in sample B, you end up, again, with just the antigen alone. In the third step, you're going to add the secondary antibody. Remember, like I told you before, indirect indicates that we're going to need another antibody that will bind to the primary antibody. Now, this secondary antibody, it has a little enzyme that's linked to the stem region. So that's where the name enzyme linked comes from, because there's an enzyme that's linked onto this secondary antibody. So if the patient had antibodies that bound to the HIV antigen, then the secondary antibody is going to bind to the primary antibody, as in case for the A. Okay, the secondary antibody bound here. Whereas for patient B, there was no primary antibody, so the secondary antibody has nothing to bind to. It's just kind of hanging out. Again, we're going to rinse the wells. So in sample A, we have the antigen, primary antibody, secondary antibody all linked up. But in patient B, we've now rinsed away all of the secondary antibody. In our final step, step 4, we're going to add a substrate that's going to interact with the enzyme that's on the secondary antibody. This substrate is going to cause a color change. So what you see for patient A is that this substrate interacts with the enzyme on the antibody and it causes the color to change. Whereas for patient B that was negative, there's no color change. So as I mentioned, color change indicates positive, no color change indicates negative. Let's talk about the second type of ELISA, capture ELISA, also known as sandwich ELISA. And you'll see why it's called sandwich, because we're going to make a little sandwich. So this test, for example, is used to diagnose measles virus. So how this works is the first step is we're going to add a known antibody. So we'll pretend like we're talking about measles. So we're going to add a known measles antibody. So last time in the indirect ELISA, we first added a known antigen. This one's the opposite. We're adding a known measles antibody. Next, we're going to add the antigen that came from the patient. So we'll say the unknown antigen. So if the patient had the measles virus and we took a sample from the patient and measles was present, the measles virus antigen would attach to the antibody. And of course, if the patient didn't have any measles virus in their system, then it wouldn't attach. Now, step 3, again, we have to add that enzyme-linked antibody or the secondary antibody. This time, the secondary antibody that has the enzyme linked to it is also going to attach to the antigen. So you're creating a sandwich of primary antibody, antigen, and secondary antibody that all sandwich together. And then finally, again, we're going to add a substrate that will interact with the enzyme on the secondary antibody and cause a color change. So again, color change would indicate a positive result. The last thing to mention is some of the tests that we use to diagnose viruses. This is really important because remember, viruses don't grow on media like bacteria do. Viruses need a living host. So it's a little bit more tricky to diagnose a viral infection. You can't just swab a sample and throw it on an agar plate. If you want to be able to grow a virus in a lab, you need either a cell culture, an actual animal to inject the virus into, and not all viruses will grow in all animals, or maybe even an embryo, a bird embryo. So if you actually did need to grow the virus, you need living cells. Most frequently, the way we will diagnose a viral infection is simply by looking at the signs and symptoms of the patient. This, for example, is showing you a patient that has herpes simplex 1, a cold sore. The best thing to do is really just go, if it's a DNA virus you suspect, you could use PCR. And if it's an RNA virus, you could use RT-PCR. Like I said, they do make some of those rapid point-of-care tests, especially like for HIV, for hepatitis C. Okay, so they have these rapid tests but I told you the problem with them is that they're not always accurate. You get a lot of false negatives and false positives. If you have a blood sample, you could also do an ELISA. Again, ELISA is very fast, rapid results, much more accurate than point-of-care tests. And if you need to verify an ELISA, you can use a Western blot for viruses. If you do need to visualize a virus under the microscope, you have to use electron microscopy, which is extraordinarily expensive and hard to do. So it's really not very common that we use electron microscopy, but it might be needed if, for example, we had a novel virus like the novel coronavirus that we needed to really examine. Another thing that could be done is cells could be taken from the patient themselves. Remember, some viruses leave behind cytopathic effects like changes in the shape of the cell. So, for example, human papillomavirus, when a woman gets a pap smear and they are screening the cells of the cervix, they're looking for any changes, so they will visualize it this way to screen for HPV. Also, some herpes viruses, they might do this. So these are some things to keep in mind, some of the techniques we use for diagnosing viral infections.